Okay, good morning, everyone. Thank you for having me here and giving me this opportunity. Today, as you can see from the title, I want to talk to you about the impairment of the sodium channel beta 4 subunit in a specific type of epilepsy. And before talking of that, I want very quickly, I don't want to bother anybody, but we need to know what is epilepsy. And as you can see in this slide, is defined as a recurrent seizures disorder that is due to abnormal electrical activity from brain. And underneath this name, epilepsy, is like an umbrella term, including a lot of different syndromes or diseases. They do all have similar characteristics and phenotypes, which are or symptoms, I would say, which are the seizures, but the phenotypic and pathological disorders are different. And the International League Against Epilepsy every couple of years is updating the classification until the very minuscule details. But in here, I've brought you the main classification of epilepsy. And we can see, you can see on your left the generalized seizures. And these are seizures involving both the hemisphere and all the parts of the brains. Whereas on the other side, we've got partial or so-called focal seizures, which are not only involving one only hemisphere of the brain, but also a specific area or focus, at least at the beginning. So in that case, they are defined simple partial. If the seizures spread to other brain regions, they are classified as complex partial. And the epilepsy that I've been working and focusing in the last many years of my research is classified as a simple partial, and is called temporal lobe epilepsy. Because it affects the temporal lobe part of the brain. And unfortunately, it's the most common adult epilepsy, and it makes up approximately half of the cases amongst all the other epilepsies. There are peculiarity about this kind of epilepsy that in the majority of the cases, the patients are affected by a pathological specific feature, which is atrophy of the hippocampus, which I've reported here. You can see this is an MRI coronal section of a brain in which the left hippocampus is much smaller compared to the right. And in here are two histological sample of post-mortem, of course, of a non-affected patient, not affected by hippocampal sclerosis, and a patient which was affected by hippocampal sclerosis. And I don't know if it's so visible that the two more affected areas by atrophy are the CA1 here and here, and the CA3 regions of the hippocampus. So this patient, unfortunately, you can see that 60% of this specific class of patients respond to drug therapy, but not all of them. 40%, unfortunately, do not. And so for them, the last resort is undergoing surgery to resect that part of the hippocampus, which is atrophic, and is the focus of the seizures. Then not all of them have got a positive outcome after the surgery, but my today's talk will be focused on these 40% not responding to drug therapy, some of which I've put down here, and they're all sodium channel blockers. Now, I'm doing a small parenthesis about a quick background, because we did some microarrays or transcriptomic studies using specimen taken from that class of patient, and we compared that with non-epileptic specimen. And this data brought up to our attention that one specific subunit of the sodium channel, which was the beta-4 subunit, had a very differential expression between the epileptic subject and the controls you can see grouped in here. And if you go down, you find the sodium channel beta-4 in the CA1 and CA3 of the hippocampus, with the very lower expression compared to CA1 and CA3 of non-epileptic samples. So it was not totally unexpected, in the sense that exactly there are a lot of anti-epileptic drugs which are prescribed to these temporal lobe epilepsy hippocampal sclerotic patients with different mechanisms which target different molecules, but quite a lot of them, as you can see here and here, which you can see better in the next one, are targeting the sodium channel, the voltage-gated sodium channels. So what is the function of this channel to be so important in regulating the excitability of the central nervous system? They've got a major role in creating action potential and therefore controlling the neuronal excitability. On the top of this, in particular, the beta subunits have other function. As you can see down here, they can modulate the gating kinetics of the whole channel. They can regulate cell adhesion and also migration and neurite outgrowth. What is the structure of these channels that also my predecessor in this morning mentioned? This is the structure of the voltage-gated sodium channels, which are made from very big alpha subunits that you can see here. It's all one subunit with different transmembrane domain, intracellular domain, with a number of modulator sites. And a lot of studies have been focused on the alpha subunits, which are 10, to design and consider sodium channels blocker. But the beta subunits have been very much neglected so far. And beta subunits are 4 subtypes named from 1 to 4. And as you can see here, they are small, very small compared to the alpha, and define auxiliary subunit. Beta 4 is much more similar in structure and amino acid sequence to the beta 2. What I think is interesting to point out from this slide is that the beta subunit have got quite a lot of their domain, which is on the extracellular part of the neurons. So they could be very good target to have newer drugs, more effective to block these channels. In the study following the transcriptomics, which were addressing our attention to this beta-4 subunit, we took again human sample. Here are the reported the human sample that have been used in this study. And we decide to verify the expression of the transcript, the single transcript for that subunit in, of course, in the epileptic TLE-HS specimen and in non-epileptic specimen. And in order to do that, we perform a quantitative real-time PCR. And we analyze them with two different, slightly different tests, the Mann-Whitney U test, unpaired, and the Wilcoxon sign rank test, as you can see here. From the first test, we found a decrease of the expression of this subunit in the epileptic sample, which didn't show any significance, whereas with the Wilcoxon test, we found a significant decrease of the subunit, which was corroborated the transcriptomes studies. At this point, we were thinking, okay, we know the messenger is downregulator or impaired. What about, is the protein following the same trend or not? And so we performed quantitative Western blot analysis, extracting the protein from the same patient on which we did the quantitative PCR. And we notice exactly the same trend, as you can see from a qualitative picture on the top left here, between the controls and the epileptic. The red band is an housekeeping gene, is a beta actin, and the green band is our subunit, the sodium channel beta 4, which is also from a qualitative point of view, it is clearly that is less expressed, confirmed by quantitative data that you can see at the bottom here, in which with different tests, we did a significant decrease of this protein subunit. What was the next step of our study? We investigated the functional implication of this difference in the sodium channel for beta 4 subunit expression. And this challenge was addressed by generating a co-expression network using publicly available transcriptomic data set from sample TLE-HS sample with the same clinical phenotype as the one used in our experiment. And these were followed by an investigation of co-expression with the sodium channel beta 4. And as you can see from these lines, we found 83 genes significantly, importantly co-expressed with the sodium channel beta 4. Unfortunately, only 77 of these genes had the functional information available on the platform. And using our information retrieved from other bioinformatic platforms such as David and Amigo 2, we have associated co-expressed genes and have identified, as you can see here, several biological mechanism components which are associated to these genes. And you can see the metabolism, genes associated with the mitochondrial function, genes associated with the nervous system main function, responses to stress, some DNA repair binding, cell adhesion, cycle, motility, proliferation, cytoskeleton. And then some genes co-express in association with potassium ion channels. So if from these we move to the hypothesis or further analysis also of these co-expressed genes. And in these slides, we report putative mechanisms which are involved in the control of expression of sodium channel beta 4. And they are associated to other sodium channels or potassium channels and calmodulin proteins and calcium channels. Where we can see the red line are targets that can influence the expression of this sodium channel beta 4. So all these genes can represent target to design or modify existing drugs to better treat those patients who do not respond to the actual sodium channel blockers. And of course, all these will improve the treatment for that 40% of patients who are not responding to the actual therapy. And this would be a benefit for the patients, first of all, their carer, their family, and for the public or private health sector, because undergoing this kind of surgery, like a lobectomy or partial lobectomy, is very expensive, is very invasive for the patient, and is not always successful and lead to some serious side effects. So the conclusion from this data is that we have confirmed that the transcript of the sodium channel beta 4 in drug-resistant patients is lower compared to the non-epileptic control specimen. Also, the protein level present a reduction in expression. Furthermore, the co-expression network of the sodium channel put to light a linkage between the subunit itself with potassium, calcium activated channel, and also the potassium 2-pore domain. With this approach, we also infer a number of other potential effector and drug target for the variation of these subunits. I would like to acknowledge my collaborators, which are Dr. Mariam Shelabi and Edward Sims from the Biomolecular Research Center at Sheffield Hallam University, Dr. Luis Takeshita, Professor Farciani from the Institute of Integrative Biology at University of Liverpool, and Mr. Dev Bhattacharya from the Neurosurgery Sheffield Hallam Hospital. If there are any questions, I'm happy to answer.
Very interesting talk. I have a question that are there any chemicals available now to modulate this potassium channel, sodium channel, and is it safe to modulate these channels?
Okay, there are drugs acting targeting the alpha subunits in which some mutations have already been reported. But nobody has ever targeted this subunit as possible drug target. We are working on molecules to do that. We are working at the present time on molecules. I'm collaborating with the chemist because that is one of our project ongoing. But I cannot share anything yet about that. We are a very preliminary stage of that research. But that is the aim, of course. I don't know if I've answered your question.
Thank you so much for your presentation. Now, floor is still open if anyone having further questions.